And welcome everyone. Thank you all so much for joining in today as we present our speaker for our UW Faculty Speaker Series. We're so excited to bring on Professor David Veesler, who will be presenting on his topic around COVID-19 variants and his research around this topic. So we're thrilled to be able to host this information. Just a few housekeeping rules. We will have our speaker today moving through the presentation topics. And as questions come up, we'll ask that folks hold off on questions to either the end of our presentation or at the designated stop points. So we do have some points during our conversation today where questions will be fielded. With that being said, I'll go ahead and turn things over to Professor David Veesler. So thank you so much.
Thank you very much for the kind introduction, and thank you for inviting me. I'm really happy to be here. I'm an associate professor of biochemistry in the School of Medicine. I'm also an investigator with the Howard Hughes Medical Institute. And my lab is interested, broadly speaking, in how viruses enter host cells and how the immune system responds to infection. And we have had a long-term interest in coronaviruses, which we have been studying for almost a decade now. And especially the spike glycoprotein, which is the infection machinery allowing entry into cells, and that is shown in red in this artistic rendering here. So I thought we could start this presentation with me sharing with you some of the work we had done before the pandemic and what was the state of the art in the field before the emergence of SARS-CoV-2. And actually, we can simplify and summarize all the work that we and others had done into a movie we put together with Janati Wasa that really captured everything we knew about coronavirus entry into host cells, or most of what we knew. We can see here spike homotrimers shown here in 3 different colors with the receptor binding domains, or RBD, that is changing conformation in this animation, allowing to adopt at least 2 conformational states at the apex of the spike. These conformational changes allow the receptor binding domain to engage the host receptors, and then the host protease can cleave the spike to activate it. The apex is removed, and the fusion machinery can mediate membrane fusion of the host and viral membrane, allowing genome delivery and the infection cycle to start. So these findings remain true. Obviously, a lot more work has been done by we and others throughout the pandemic, but I thought it was a good way to bring everybody up to speed. So if we go back to the end of 2019, in the last few days of 2019, I remember when we were hearing reports of an outbreak of pneumonia of unknown origin. And some of these cases were described by doctors in Wuhan, China, as similar to SARS, which was a disease mediated by SARS-CoV-1, which emerged in 2002 in China as well. And I recall how our work started. I recall vividly this Friday afternoon, January 10th, 2020, around 5 p.m. Pacific time, the sequence of the very first SARS-CoV-2 genome was released, and that really launched our work and the one of others. And one of the first questions we decided to ask was, what is the receptor used by these newly emerged viruses to enter host cells? And we designed genes to be able to answer this question. And Lexi Walls in our lab showed that transfecting cells with ACE2, which is the receptor used by SARS-CoV-1, and which we suspected could be a receptor for SARS-CoV-2 based on sequence identity between the 2 viruses. And Lexi showed that transfecting ACE2 into cells rendered them permissive for spike-mediated entry, establishing ACE2 as a bona fide receptor for this newly emerged virus. That was actually done before the virus was even called SARS-CoV-2. Young-Jun Park wanted to understand more in depth the mechanisms of viral entry and determine cryo-electron microscopy structures, which is a method allowing molecular imaging at high resolution of the spike glycoprotein, which really is the infection machinery, as I showed in the previous movie. And so Young obtained multiple structures which are snapshots of this infection machinery, forming homotrimers with each protomer shown in a different color. And the first snapshot revealed that the receptor binding domains, which I will be calling RBDs for the rest of my talk, were in a conformational state which we describe as closed and packed along the rest of the spike glycoprotein. We also detected the presence of a different snapshot in which one of these RBDs had an open conformational state, which allows exposure of the receptor binding site and engagement of ACE2, which we had just shown to be a receptor. So these findings showed very early on in 2020 that these conformational changes recapitulated the ones I showed in the movie that we knew existed for SARS-CoV-1 or MERS-CoV, for instance, and really highlighting that this newly emerged virus was using a similar mechanism of entry into target cells than previously known coronavirus pathogens. These structures have been used as a blueprint by literally thousands or tens of thousands of labs by now to map mutations that they emerge in variants and understand their effect, as well as to guide the design of vaccines and therapeutics throughout the pandemic. So we didn't know a lot about antibody responses to coronaviruses before the COVID-19 pandemic, but we and others had done some work that led us to expect a few things in terms of the targets of antibodies on the spike glycoprotein. We knew that the RBD were the target of potent neutralizing antibodies against SARS-CoV-1 and MERS-CoV. Some of them we characterized a few months before the emergence of SARS-CoV-2. And we expected the N-terminal domain of the spike, which we also call the NTD, to be the target of neutralizing antibodies, especially based on the work that was done on MERS-CoV. These two domains are part of what we call the S1 subunit, which really has for role to mediate attachment of the virus to target cells. Whereas the second part of the spike, this is proximal to the membrane, is the fusion machinery, which is also called the S2 subunit, which is the C-terminal half of the molecule, and that is in charge of mediating fusion of the virus and host membrane to allow infection to be initiated. So at the beginning of the pandemic, and actually even before that, we established a close partnership with Davide Corti's group at Vir Biotechnology. And we set out to jointly characterize antibody responses in infected individuals, COVID-19 patients initially, and then in vaccinated individuals or people that had acquired hybrid immunity, which corresponds to a mixture of vaccination and infection. So one of the things we observed when cloning monoclonal antibodies from the memory B cells of various individuals, here we are looking at 3 individuals, each circle corresponds to a single subject, and the percentage we are seeing correspond to the number of monoclonal antibodies that were cloned from their memory B cells that are specific for the RBD, the NTD, or other regions of the spike. What's pretty obvious is that the RBD accounts for most antibodies that are targeting the spike in infected individuals. But a non-negligible fraction, which is variable across individuals, target other domains such as the NTD. And in the 3 individuals, we can see that this fraction varies between 6 all the way to 20%. So Matt in our lab set out to study antibody responses to the SARS-CoV-2 NTD early 2020, for which we didn't know anything at the time. And using a combination of cryo-electron microscopy, which again is an approach allowing high-resolution molecular imaging, and combining that with binding assays, Matt delineated the presence of at least 6 distinct antigenic sites at the surface of the spike NTD. Here, I'm illustrating 4 such antigenic sites with antibody Fab fragments colored differently at the surface of the spike, which is shown in gray. So one of the observations we made very early on was the fact that all potent neutralizers of SARS-CoV-2, which targets the NTD, are actually binding a single site, which is highlighted by the Fab fragment in red. And we therefore call that site an antigenic supersite, which appeared to be the target of all NTD-directed neutralizing antibodies with potent activity against SARS-CoV-2. And it turns out that the most potent NTD antibodies are comparable to best-in-class RBD-directed antibodies, which I will be describing in the next slides. To evaluate the ability of NTD-directed antibodies to protect against SARS-CoV-2 challenge, we used the hamster model of infection and administered an antibody targeting the site shown in red 1 day before challenge. We call that prophylactic administration. And we evaluated administration of 2 doses of the antibody, 1 mg per kg and 4 mg per kg, which we compare to a control group, which are hamsters receiving a different antibody, which is not directed at SARS-CoV-2. What we can see is that administration of an NTD-directed antibody, which is a potent neutralizer in vitro, led to protection against viral replication in the lung of hamsters in vivo, as shown here with most animals. Each animal is represented by a dot. Most animals were being at baseline levels. However, as you can probably see, a few animals did not appear to control viral replication in their lungs and were not different from the control group. So we were curious to understand what could be happening there. And we decided to sequence the virus in the lungs of these animals that were not controlling SARS-CoV-2. And what we realized that they had acquired various mutations, and they all had in common a deletion of residue 144, as shown here for the 2 mutants, as opposed to the control group, which is the same sequence as the ancestral virus, which has a residue Y, a tyrosine residue. It turns out that position 144 maps in the antigenic supersite, and this deletion leads to knocking out binding and neutralization of NTD antibodies targeting these sites. What is even more interesting and fascinating to me is that in early 2021, the alpha variant, which was globally dominant, also acquired a mutation different from the ones in the viruses we sequenced, but that also deleted residue 144. So that showed that we recapitulated in the hamster model of infection the viral evolution that was going on in the real world at the same time. So it turns out that the NTD is mutating very rapidly. And we can see that by comparing sequences that are deposited all over the world of viruses for the past 2 years. So by determining cryo-EM structures of key SARS-CoV-2 variants, Matt was able to show that this rapid accumulation of mutations in the NTD leads to remodeling of the antigenic supersite, as shown here. And this remodeling can be as marked as refolding from a beta strand configuration to a helical configuration. But at the end of the day, these are all distinct molecular solutions to the same problem, evading neutralization mediated by NTD antibodies. So this rapid accumulation of mutations is highlighting that it is very difficult to target the NTD for development of countermeasures because antibodies elicited by infection or vaccination with one variant and targeting the NTD will not work against the next or the previous variant. At the same time, Young and Ali in our lab were characterizing antibody responses against the SARS-CoV-2 RBD. And using cryo-EM and binding assays, we delineated an antigenic map of the SARS-CoV-2 RBD, which here again is illustrated with Fab fragments of different colors bound to the spike glycoprotein in gray. So this work was also done in close collaboration with David Cortes' group at Vir Biotechnology, which allowed us to define at least 4 antigenic sites at the surface of the spike. Some of these sites are so large that we are dividing them in subsites. Here I'm showing you 3 different subsites within antigenic site 2. And they are covering almost the entire surface of the RBD And by integrating structural and functional data, we're also able to understand the determinants of neutralization potency directed at the RBD, as well as neutralization breadth, which means against various variants and even a larger group of viruses, which we call sarbecoviruses, comprising SARS-CoV-2, SARS-CoV-1, and many viruses found in wildlife, especially bats. In the interest of time, I will describe only one, or I will highlight only one of the antibodies we worked on. And this antibody was actually isolated early in 2020 from the memory B cells of a survivor of SARS-CoV-1 infection back in 2003. This antibody neutralizes SARS-CoV-2 and SARS-CoV-1 because it was isolated from a SARS-CoV-1 survivor of infection. And the antibody has been used as a therapy for patients at risk of developing severe COVID infection upon infection since early 2021 worldwide. The antibody is called sotrovimab, and we're really, really excited to have been able to work hand in hand with Vir Biotechnology to reach that outcome. And that's the very first molecule that the Veesler lab brought to the clinic. So what became very interesting is that because this antibody was not isolated from a survivor from an individual infected with SARS-CoV-2, but instead infected with SARS-CoV-1, the sites recognized is highly conserved, which we showed by structural study. And what that means, practically speaking, is that when variants emerge, they are highly unlikely to mutate at that site, which is conserved throughout viral evolution across two widely divergent viruses, SARS-CoV-2 and SARS-CoV-1. So when Omicron emerged, which I will be describing in a few slides, but I wanted to highlight that when Omicron emerged at the end of 2021, and the first Omicron sublineage that became dominant was called BA1, we showed through structural studies and functional studies that were led by Matt in our lab, that out of the 15 RBD mutations accumulated in the Omicron BA1 RBD that are shown in red here, as compared to the ancestral virus, only one of them was mapping in the epitope recognized by sotrovimab. So the antibody avoids most of them. And that one mutation, which is a glycine to aspartate mutation at position 339, is accommodated by the antibody, as shown by our structure, which also explains that at the time in November, December 2021, when Omicron emerged, sotrovimab was almost the only antibody that did not suffer major reduction in neutralizing activity in vitro against this new variant as compared to all other clinical antibodies. So then we decided that we understood that there were so many antibodies targeting the RBD, but we were wondering, what is their quantitative contribution to neutralizing activity upon infection? To answer this question, we depleted the plasma of infected individuals from polyclonal antibodies directed at the RBD. And we asked, how much neutralization is left when we do that? We realized that we obtained a tenfold reduction in neutralizing activity when removing RBD antibodies, RBD-directed antibodies. And that really underscored that the RBD is not only immunodominant, but it is also the main target of neutralizing antibodies in the plasma of previously infected individuals. So based on this data, we decided to design a vaccine using an approach which is completely different from the one used by Big Pharma. What we thought is that we should focus antibody responses on this key domain of vulnerability, which we had just identified, the RBD. Whereas the 4 vaccines that are distributed in the US and many more worldwide are using the whole spike. So we knew that using the RBD only, since it is a small domain, will lead to moderate, at best, immunogenicity upon vaccination. And to overcome this challenge, we decided to display it as a multivalent array at the surface of a 2-component protein nanoparticle that was designed by Neil King's group in our department. And I really want to emphasize this fantastic collaboration we have had with Neil's group throughout the pandemic and even before, and we worked hand in hand to develop coronavirus vaccines. And this work was led by Lexi Walls in our lab and Brooke Fiala in his lab, which led to designing genetic fusions of the RBD through genetic linkers to one of the two components of the nanoparticle we decided to use to generate the vaccine. The second component of the nanoparticle can be added afterwards, and that leads to assembly of this icosahedral nanoparticle displaying 60 copies of the SARS-CoV-2 RBD at its surface in a highly flexible array. Using electron microscopy, we confirmed that we had assembly to the target geometry, and you can even see at the periphery studded RBDs protruding as designed, which is the vaccine we ran with for preclinical evaluation. In a preclinical study in mice, we showed that 2 doses of various flavors of the nanoparticles shown here led to neutralizing antibody titers that were an order of magnitude higher than those obtained by vaccination with 2 doses of the prefusion stabilized spike, which is the engineering strategy that was used for all 4 vaccines used in the US. We showed in collaboration with the Berwick lab at the University of North Carolina that mice that received 2 doses of nanoparticle were protected against weight loss upon SARS-CoV-2 challenge, as shown here, which was not the case for unvaccinated mice. And in collaboration with the Phylogeny-driven lab at Stanford, we showed that rhesus macaques that received 2 doses of the vaccine were also protected against SARS-CoV-2 challenge, which is illustrated here with PET scan of the lungs of these animals, showing that whereas unvaccinated macaques suffered lung inflammation, as shown here, vaccinated animals using one of the 2 clinical adjuvants shown here led to protection from signs of lung inflammation. So which are really, really exciting data. So obviously, while we were doing all this work, as I briefly mentioned a few slides ago, at the end of 2021, we entered a completely new phase of the pandemic with the emergence of Omicron, the accumulation of a staggering number of mutations in the genome of this virus compared to previous variants, and especially the spike glycoprotein that accumulated 37 amino acid mutations as compared to the ancestral virus was completely unprecedented throughout the pandemic. So we set out to evaluate in real time the impact of the mutations found in these variants, which are all named Omicron, but it's actually a very, very diverse family of variants. And we define them as sublineages in the absence of better terms. And Amin in our lab decided to try to understand from a functional standpoint, what are these mutations doing to the function of this spike, which is to mediate entry into cells. And the first step is that to answer this first question, the first step, Amin developed a membrane fusion assay in the lab, which is a GFP-based fluorescence assay, allowing to monitor live cell fusion kinetics with the spike expressed at the cell surface, mediating fusion with target cells and leading to reconstitution of the fluorescence signal through GFP. We are going to be watching 4 different movies at the same time, corresponding to fusion mediated by the ancestral spike glycoprotein, the delta spike glycoprotein on the top right, the Omicron BA1 spike glycoprotein. This is the very first lineage that became globally dominant at the end of 2021, early 2022. And the BA5 spike glycoprotein, which is the lineage that was globally dominant a couple of months ago worldwide. So what this movie shows is that the delta or the ancestral spike mediates much more extensive and faster fusion than Omicron sublineages, as shown here, indicating an impairment in fusionability of the spike. And that was an unexpected finding. And at the same time, we also showed that the entry route of Omicron BA1 had changed compared to previous variants. And as opposed to fusing with the plasma membrane, Omicron BA.1 was entering through endocytosis and fusion with the endosomal membrane. Another important aspect of viral entry is the ability of these variants to interact with the host receptor. And presumably, the higher the affinity, the better it is for the virus, for two reasons. One is because it leads to better interaction with the target cells. But the other one, which becomes really clear, is that the accumulation of a large number of mutations increasing affinity for the receptor allows to accumulate other mutations that are evading antibody responses, serving as a buffer that will, and these mutations can affect negatively ACE2 binding affinity. So there is a balance, and these viruses can acquire a lot of mutations. And as I just mentioned, that can lead to enhanced ACE2 binding affinity, which is the case as shown here with two different methods for all Omicron sublineages as compared to the ancestral virus. But the advantage, besides just interacting with the receptor, is that it then allows to accumulate even more mutations that can be deleterious for receptor engagement. But the increase of affinity was so high that it is still good enough to mediate infection. And I think this is best illustrated by the next panel I will be showing you, in which we evaluated how primary vaccination, which meant two doses of all vaccines except for the Johnson & Johnson, which was a single dose, did not lead to strong neutralizing activity against Omicron. So this is a very complicated plot. Let's start by breaking it down by colors. So the first color, the green color, corresponds to previously infected individuals, and all other colors correspond to individuals that received 2 doses of various vaccines, the Moderna, the Pfizer, Novavax, a single dose of J&J, and 2 doses of AstraZeneca, Sputnik, and Sinopharm. So these are pretty much all vaccines, and definitely all vaccine platforms you can find worldwide. And what's important is that now, if we look at the first column of each group, which is vaccination, which is neutralization against the vaccine matched virus, we see we have pretty reasonable titers that vary across vaccination platforms, but they are pretty reasonable for all vaccines and even decent upon infection. However, if you look at neutralization against any of these Omicron sublineages, you see that we have marked decrease, and in many cases, there is nothing left against Omicron sublineages. And that illustrates the impact of all these mutations that are really mediating immune evasion. And that was obviously a big problem when the virus emerged. The good news is that administration of booster doses, and here we are looking at 3 doses for all these vaccine platforms, except Johnson & Johnson, which was a single dose during the primary vaccine series, and therefore a second dose was administered as a booster. Neutralization of all these variants became much, much higher upon administration of the vaccine booster, which explains why we recovered a reasonably high protection against severe disease and hospitalization, but unfortunately not so much against infection and transmission. So you're probably wondering, how is our vaccine, the RBD nanoparticle, faring against Omicron? And so we had the opportunity to evaluate that in mice in collaboration with Deb Fuller's lab here at the UW, and in rhesus macaques in collaboration with Barry Pulendren's lab at Stanford. And I'm just showing you here the non-human primates data showing that 3 doses of the vaccine also elicit much, much higher titers than 2 doses against Omicron. And that's the difference between the ancestral and the BA.2 Omicron neutralization. And upon challenge, we see that 3 doses of the vaccine completely protected against viral replication in the lung of these animals. So the vaccine actually entered clinical trials a while ago and was approved for use in humans in South Korea in June. And now our partners at SK Bioscience are seeking WHO approval. And since the vaccine does not require deep freezing for storage, and the University of Washington is licensing it royalty-free for a possible distribution through COVAX, we are hopeful that we can help meet the global demand for doses, and especially people in low and middle income countries, where we still have 2 billion people that have not received a single dose of vaccine. So one of the questions that we were wondering, and that many people are probably wondering, is how does it work that you can keep immunizing with the same vaccine? As of a few weeks ago, we have a new bivalent boosters in the US, but even last month, we were still using the same vaccine, the same sequence of the ancestral virus. And how is it possible that you keep immunizing with a vaccine sequence that is very different from the virus circulating, and that despite that, we elicit neutralizing activity to reasonably high level and protection against severe disease? So to answer this question, we decided to analyze the specificity of memory B cells in various human cohorts towards the RBD of the ancestral virus and Omicron RBDs. In this plot, I'm showing you the magnitude of binding against the ancestral RBD on the x-axis and the B1 RBD on the y-axis. And each dot represents the supernatant, therefore the antibody present in a culture from a single or an oligoclonal culture of memory B cells. The red dots show the memory B cell supernatants that were able to block interaction of ACE2 with the RBD, which is a correlate for neutralizing activity. If we look in the top right quadrant, we can see that 3/4 of memory B cells are actually cross-reactive with both the Wuhan and the BA1 RBD. And we are here looking at a cohort of individuals that were infected in Washington state in early 2020 and then vaccinated 2 or 3 times. And we obtained the exact same plot with individuals that were vaccinated 3 times. Both cohorts were exposed to a single type of spike sequence, which is the ancestral spike sequence. If we look at breakthrough cases, these are people that were vaccinated 2 or 3 times before experiencing a breakthrough infection with Omicron BA1 in Washington state. We can see the plot looks quite similar, although we have an enhanced proportion of cross-reactive memory B cells compared to the previous cohorts. We also had Wuhan 1 ancestral specific memory B cells, as was the case here, but we do not see the apparition of Omicron specific memory B cells, and that holds true up to 6 months past infection. So what these data are showing us is that exposure to a highly divergent spike, such as the Omicron BA1 spike in the case of a breakthrough infection here, primarily recalls existing memory B cells that were elicited by vaccination, but did not elicit new memory B cells, at least in the cohort we have been able to analyze. And this is a phenomenon called immune imprinting, which tells us that exposure to a single distinct spike once might not be enough to trigger the formation of new memory B cells, at least within the time frame we analyzed. And we may need to wait longer or a second exposure through vaccination to a distinct spike to elicit new memory B cells that will be specific against the new variants. The fact that we are eliciting cross-reactive as opposed to specific memory B cells is not really a problem. Recalling these memory B cells that are cross-reactive tells us that they are binding to the new variants, and a fraction of them will be neutralizing, explaining why you can keep vaccinated with the same sequence and eliciting neutralizing activity. So this is not to say that it's not good to be exposed to a new spike. It is just what we have observed so far, and we obviously encourage everyone to use the bivalent booster, which I did myself 2 weeks ago. So because the RBD is so important for vaccine match neutralization, John in our lab decided to understand the contribution at the quantitative level of RBD-directed antibodies upon vaccination. First, by depleting RBD-directed antibodies from the plasma of vaccinated individuals, John showed that vaccine match neutralizing activity was decreased about tenfold, which is exactly what we observed for infected individuals, suggesting that it holds true independently of the exposure type. NTD antibodies had a small but statistically significant contribution to neutralizing activity against the vaccine-matched virus. However, when looking at cross-variant vaccine mismatch neutralizing activity. We saw that depletion of RBD-directed antibodies completely abrogated neutralizing activity of variants. Here we're looking at the Delta and here the BA1 Omicron variants. And the NTD antibodies did not have any contribution to polyclonal plasma responses. So that's absolutely fascinating. And that illustrates what I was telling you early in my talk, that the NTD mutates very rapidly and that antibodies elicited by one variant are not useful against other variants. And this work resonates very well with the work we did with David Cordy's group to isolate and characterize monoclonal antibodies that were cross-reacting and cross-neutralizing a wide range of variants. I'm showing you here 4 structures in this composite model in 4 different colors of antibody fragments of which we call broadly neutralizing sarbecovirus antibodies. What this means is that these antibodies can actually neutralize a wide panel of variants. The first 4 columns correspond to the 4 antibodies shown here, and each of these different symbols correspond to one of these many variants. And you can see that most of them neutralize equivalently almost all variants, but not all of them, but almost all of them. And sotrovimab, for example, I was telling you, neutralized the BA1 variant with a 2- to 3-fold decrease in neutralizing activity. And this decrease was higher against BA2 due to further changes. And while doing this work, we found a new antibody, S2X324, shown here, which hardly suffers any changes upon mutations found in variants. Neutralizing activity remains very, very high in the single-digit nanogram per ml. And we decided to understand the mechanism of resilience to the emergence of variants, and that's work done by Youngjun Park in our lab. And a structure of X324, this antibody bound to the SARS-CoV-2 spike, revealed, as shown here, that it binds to an epitope on the RBD comprising residue 440 to 450 that is hindering subsequent binding to ACE2 through steric hindrance, but also protecting in vivo in the hamster model of infection at various doses independently of the use of Fc-mediated effector functions. So that's really, really exciting. And this is what we call pan-variant neutralizing antibodies, because it neutralizes absolutely all variants that are circulating out there. However, I want to caveat that statement with the fact that if SARS-CoV-2 has taught us one thing, it taught us many things, but if it has taught us one thing, is that there is no single countermeasure that an RNA virus cannot escape. It is true that this antibody has pan-variant neutralizing activity, but it turns out that SARS-CoV-2 keeps evolving because this is what viruses do, especially RNA viruses. And there is some work that was done by Mark Johnson at the University of Missouri looking at cryptic lineages found in sewage water that identified some mutations that I found absolutely fascinating, because these mutations were very similar to the ones we identified as possible escape mutants from that quote-unquote pan-variant antibody. And more recently, these mutations were not found in cryptic lineages, but they were found in the main lineages that are starting to emerge all over the world. It is a really, really complicated figure. And you can see that Mark Johnson, who developed that figure and posted it on Twitter, said it's going to be difficult to keep track with all these variants. The reason I'm showing this figure is twofold, is that one of them, the position 444, which is the position of escape from this antibody that I just showed you, is emerging in pretty much all of the variants that are on the point of becoming established everywhere worldwide. But all these variants, we are looking at very different evolution from what we have seen so far, because as opposed to seeing a new variant coming out of nowhere, meaning a very long branch of evolution, here we see that all these variants are descending from BA2 and are accumulating pretty much mutations at the same 5 positions that are listed here. One of them is the escape position that is key for this antibody. And we have a convergent evolution, which we have not really seen before in the pandemic, which makes it very, very difficult to predict what will be the next variants to become globally dominant. I would like now to take a few minutes to tell you about the work we are doing to develop vaccines that could elicit broader immunity against sarbecoviruses, because it has been more than 2 years that I've been saying that chasing variants is probably not something we're going to be successful at, just due to the ability of this virus to evolve. And I'm going back one slide just to show you that again. It's just staggering. We are never going to be able to react rapidly enough to variants. So it would be better to anticipate by developing strategies that are as variant agnostic as possible. But so how should we go about developing such a vaccine? So what we thought is that a pan-sarbecovirus vaccine or vaccine eliciting broad sarbecovirus immunity should be able to target antigenic sites that are conserved across a wide diversity of sarbecoviruses, and therefore that are conserved throughout sarbecovirus evolution and are less prone to mutate, perhaps due to their key role in the structure and the function of the spike glycoprotein. So this is a cladogram showing the classification of sarbecoviruses based on the sequence of their RBDs, which again is the main target of neutralizing activity and the sole target of cross-variant neutralizing activity. We knew that 2 clades out of these 4 clades, the SARS-CoV-2 clade and the SARS-CoV-1 clade, comprise viruses capable of using human ACE2. And some in our lab teamed up with Tyler Starr in the Bloom lab to show that clade 3 viruses, for which we hardly knew anything, were actually capable of using 2 types of bad ACE2, Rhinolophus affinis ACE2, to enter cells expressing that specific ACE2. And here I'm showing BTKY72, which is one member of this clade 3 sarbecoviruses. And Jimin, a student in our lab, just made similar findings with another clade 3 sarbecovirus. Even more concerning is that if we introduce 2 amino acid substitution in the spike of these clade 3 sarbecoviruses, we can observe in a non-replicating pseudotype virus assay that it allows entry into cells expressing human ACE2. And that brings 3 out of 4 sarbecovirus clades capable of using human ACE2. So we are using that as a blueprint to design a new generation of vaccines using a phylogeny-driven approach that will allow to target as many of these clades as possible. And as a reminder, the broadly neutralizing sarbecovirus antibodies we identified are neutralizing not only variants, but also a wide range of sarbecoviruses. Sotrovimab covers clade 1b and 1a, as is the case for S2K146, whereas S2X259 and S2H97 can cross-react with sarbecoviruses from all 4 clades and neutralize all viruses for which we have an assay. Because we do not know what the receptor is for clade 2, we can only probe cross-reactivity, but not neutralizing activity at the moment. One caveat here again is that although these sites targeted by broadly neutralizing antibodies are highly conserved across sarbecoviruses, and that's really the engine of our approach, it is important to realize that mutations and changes are always possible. It is highly unlikely that all the sites can change, but Omicron has taught us that these sites can be altered in some ways. Antigenic site 2, which is shown here on that side of the RBD, is remodeled in the BA1, and even more so in the BA2 Omicron RBDs shown in blue and gold respectively. And we showed that it dampens binding and neutralizing activity of one of these broadly neutralizing antibodies. And the large conformational change occurring here in this helix is actually also incompatible with the position of the glycan, such as when sotrovimab is recognizing the RBD, which explains why there is a reduction in neutralizing activity for sotrovimab against BA2. So it does not invalidate the strategy, but it tells us that we need to have an as diverse polyclonal antibody responses as possible, specifically targeting these broadly these sites that are conserved and targeted by broadly neutralizing antibodies. And to do that, this is work led by Lexi Walls in our lab. As I mentioned, we are using a phylogeny-driven approach, which consists in including the receptor binding domains of one representative member of each of these 4 sarbecovirus clades in a mosaic nanoparticle with goal of preferentially eliciting broadly neutralizing antibodies by displaying distinct RPGs next to each other, which only share in common these highly conserved sites, but that have widely diverse immunodominant sites that are mutating rapidly. Because we do not know what the receptor is for clade 2, and its likelihood of cross-species transmission of humans appears to be low at the moment, we also made a trivalent nanoparticle in addition to the tetravalent nanoparticle in which clade 2 is excluded. In collaboration with the Berwick Lab at the University of North Carolina and Neil King's group, of course, with whom we have worked, as I mentioned, throughout the pandemic to develop vaccines, we showed that broadly cross-reactive responses are elicited by both the trivalent and the tetravalent nanoparticle, as shown here by flow cytometry in collaboration with Tyler Starr in the Bloom Lab at Fred Hutch. There's a lot of data here, and so I just want you to take home that the trivalent nanoparticle shows dark blue colors against these 3 clades, as is the case for the tetravalent nanoparticle. But there's a distinction between the 2 nanoparticles is that only the tetravalent nanoparticle elicits broadly reactive responses against the clade 2 viruses, and not the trivalent nanoparticle, which is the one that excludes the clade 2 RBD from the formulation. So that tells us that if an antigen or a representative member of a clade is not present in the virus, in the vaccine, we may run into troubles to elicit neutralizing activity against this clade of viruses, although this is something we are still studying in mice and non-human primates. We showed that 2 doses of these vaccines elicit vaccine match and vaccine mismatch neutralizing activity against the ancestral virus. We are looking at SARS-CoV-2 vaccine, or the tetravalent and trivalent nanoparticles. And really the advantage of the mosaic kicks in when we look at neutralization of divergent sarbecoviruses such as SARS-CoV-1, for which we hardly detect neutralizing activity for a SARS-CoV-2 vaccine, which holds true for any SARS-CoV-2 vaccine out there, whereas the trivalent and the tetravalent nanoparticles comprise the SARS-CoV-1 RBD in the formulation, and therefore elicit neutralizing activity, which holds true for 2 clade 3 sarbecoviruses. We showed that vaccinated mice were protected against challenge against a highly divergent vaccine mismatch sarbecovirus, SHCO14, that shares only 82% amino acid sequence identity with the RBD of SARS-CoV-1, which is the most closely matched RBD in the formulation, as shown here for the trivalent and the tetravalent nanoparticles that did not lose weight, which is not the case for unvaccinated animals. And because I see that we are getting close to the top of the hour, and I would like to make sure we have enough time for questions, I think I'm going to stop here and take a few seconds to really thank all the wonderful members of my lab that worked with me throughout the pandemic to make all this work happen. I mentioned and highlighted many of them through my talk, but I really wanted to make it clear that it takes a village, and everyone really was a key player to make this possible. I also want to acknowledge our wonderful collaborators, and we have so many collaborations that's been fantastic to be able to interact with so many fantastic scientists worldwide. I really highlighted 2 of our key collaborations with Neil King's group in our department here in biochemistry for vaccine development, and with David Cortes' group at Vir Biotechnology for antibody discovery and characterization. With that, I would like to thank you for your attention, and I would be more than happy to answer any questions.
Thank you so much, Professor. Wow, it is a lot of information to put in an hour, but we're so glad to have been able to have you do so. One of the first questions that that was sent in the chat early on was what makes some parts of the virus highly mutagenic, whereas other parts are more stable, such as the injection machinery?
That's a fascinating question. So I think the injection machinery refers to the fusion machinery, which is the S2 subunit and really the part that is mediating membrane fusion. So I think it's twofold. One of them is the function of the fusion machinery is more constrained, and it is identical in all coronaviruses. It is to fuse the viral and host membranes, whereas the S1 subunit comprising the receptor binding domain and the N-terminal domain, either or both domains can be involved in receptor recognition depending on which coronavirus we're considering. You have to allow for some variability because different coronaviruses are recognizing different receptors. That's only part of the explanation. The other one is that, as I showed, the RBD and the NTD are completely immunodominant. The wide majority of antibodies target the RBD or the NTD. And I'm actually going to show a slide that I did not show in the interest of time, but that John did some depletion experiment from plasma obtained from vaccinated individuals. And if we deplete the antibodies targeting the S1 subunit, we can see that we completely remove neutralizing activity. It's the same as if you were using the whole spike. If we use the S2 subunit, which is the fusion machinery, the object of this question, whether it is in the pre-fusion or in the post-fusion state, there is hardly any effect that can be detected at the polyclonal level of depletion of such antibodies. And we have isolated monoclonal antibodies targeting the fusion machinery. The problem is that they are so rare that their quantitative contribution to the bulk polyclonal plasma antibody responses is hardly detectable. And therefore, there is hardly any selective pressure on the spike to change that. So the combination of functional constraints and lack of selective pressure due to very, very rare targeting at the polyclonal level explain the difference of in the rate of accumulation of mutations. I hope that answers the question.
Mm-hmm. Another question we received, are there any other receptor binding of virus besides ACE2?
Yes. Well, it depends if the question is about SARS-CoV-2 specifically or coronaviruses more broadly. Maybe I will answer in a two-tiered way to make sure I cover everything. So many molecules were proposed as alternative receptors throughout the pandemic, and we tried to characterize all in the lab, besides ACE2, of course, whether they are ancillary receptors, attachment factors facilitating ACE2 engagement, or whether they are alternative receptors. I have to say that we have had a hard time recapitulating most of the published literature on this data. We have found that host lectins, such as Siglec, appear to affect the neutralization pattern of some antibodies and could potentially participate in entry by recognizing glycans on the spike. This is a very indirect way of detecting it because we saw, again, the neutralizing activity of some antibodies was actually modulated by the presence or not of Siglec. And so it is possible that these lectins play a role, but at best, it's a minor role compared to ACE2. It has also been proposed that heparin and various carbohydrates, including sialic acids as well, or psilocides, were involved in attachment or recognition. And there is one convincing paper that was published a few months ago in Science, where they demonstrated attachment of sialic acid to the N-terminal domain. The functional relevance needs to be investigated further, but what was shown is that this sialic acid binding site was present in the ancestral virus, but not in subsequent variants. And I'm actually just remembering what I'm answering last week. Another paper showed recognition of a different psilocybes. So bottom line is that, well, ACE2 seems to explain most of what we know about the virus, but not all. And some candidate receptors were proposed, again, as ancillary receptors, attachment factors, perhaps sometimes as alternative receptors, but the data is not always reproducible. And otherwise, other coronaviruses are using a wide range of receptors. But the very interesting observation is that most coronavirus receptors are actually transmembrane host proteases. The actual enzymatic function of the protease is never involved in entry as far as we know and as far as was probed. But surprisingly, not a wide diversity, but 3 to 4 different host proteases mediate entry of pretty much all coronaviruses. There's a subgroup that uses psilosides for entry exclusively or apparently exclusively, but that's an interesting observation and for which we don't have a good explanation yet.
Thank you. Yeah, we do have some time left for any additional questions, if folks have it. All right.
Oh, I see a question just popped up. Does weight loss correlate with infection as shown in one of last slides? So weight loss is just one of the parameters we are following. So what we like following is weight loss, viral replication in the nasal turbinates and in the lungs. And the reason for that is that you can be protected against severe disease such as weight loss, but not against infection. And that's exactly what we see for vaccinated individuals in the real world. Is that again the protection of vaccines against the ancestral virus after 2 doses was just truly outstanding. It was 95% protection for 2 doses of the Pfizer or the Moderna vaccine at the end of 2020 against symptomatic infection. Unfortunately, with variants, the protection against infection has been reduced, but fortunately, the protection against severe disease and hospitalization has remained strong, especially upon administering boosters. So looking at these different parameters, weight loss, viral replication in the nasal turbinates and viral replication in the lungs, we can tease out these differences. Here, in the interest of time, and to avoid cluttering the slides, I showed weight loss, which is an easy metric to look at, but we are definitely following all these parameters.
Incredibly fascinating stuff. Mm-hmm. Right. Well, thank you, Professor, so much for this educational and informative hour together. We'd love to hear about any upcoming research or ways we can support or contact you if there is anything you'd like to share in the upcoming year. That we would love to be able to hear from you and find ways to support your work and your research as well as your team.
Well, thank you very much, and maybe I can take this opportunity to announce that we are always recruiting. Today, tomorrow we'll be recruiting, and we are recruiting people with a broad set of interests and skill sets, whether you're interested in biochemistry, in immunology, in biology, and even in genomics. So if you're interested in participating in this work with us on SARS-CoV-2, coronaviruses, and actually many other pathogens, feel free to reach out and let's talk. Thank you very much for your attention once again, and